We'll get away. Well, thanks for attending, everyone. Good afternoon and welcome. Thanks very much for making the time. I know 2 o'clock on a Tuesday is a fairly big deal, so it's genuinely appreciated. My name's Steve Lorimer and I'll be facilitating today. We have a fair few on, which is great to see. We have people joining us from universities, state agriculture departments, industry research programs, and a fair few agronomists and consultants as well. So just a few quick housekeeping items before we start. I should let you know that we're recording today's session, so no need to use an AI note taker or whatever. We'll make it available afterwards. If you'd rather not appear, that's fine, because your camera and microphone are already off, so we don't need to worry about that. Because there's so many people on the call, I've made the decision to mute everyone to keep the audio clear, but that's not to stop anyone from contributing. But what we'd like to do instead is to use the chat. So if something occurs to you during the session, you can drop it straight into the chat as it comes to you. Don't sit on it, because I'll be watching the chat throughout and interrupting Ross as we go. He gave me permission to interrupt him, which is cool. We've blocked out until about 3:30, but I expect we might be done in around about an hour, depending how many questions we get. If you need to go earlier, that's fine. Just stay in touch and watch for the video or whatever later on. So just a quick word on Agriva and why we're doing this. Agriva distributes Actinovate in Australia, and we're running today's session in partnership with Vikineswary, who manufacture it. I want to be upfront about what this session is and isn't. It's not a sales pitch. We flagged in the invitation that it is purely about the technical side of it. So what we're trying to do is put the technical picture in front of people who are qualified to interrogate that. Actinovate has a 30-year evidence base behind it. Some of that evidence is strong, some of it's mixed, and some of it Ross will explain as we go along where and when it may or may not fit, and you'll hear all that from Ross. Unfortunately, biologicals often get a bad rap in this industry. They can either be oversold or dismissed out of hand. Neither situation is useful, so the main aim today is to give you some of the mechanisms and methods and some actual data, and let you develop a greater understanding of what's been done and how it might fit into your plans moving forward. One important note before I hand over, but I just got to keep it short. Everything we cover today is the science, the mechanisms, the research, and the trial data. None of it should be taken as a recommendation for use. Labels are different between countries, and in some cases, in Australia's case, in between states, and that label applies where, so you need to apply that label based on where you are. Please read it, make sure you understand it and follow it. If something you hear today sounds relevant to your situation, that's great, but check it against your label and regulations before you act on it. So that's how we had to put that one in there. Now, presenter introductions to our presenters, and it's worth noting that both are joining us today from Kuala Lumpur, where it's actually a public holiday, so kudos to them both. That's right, Ross? Yep. Brownie points, big brownie points there. So Dr. Ross Gilmour is a consultant. Now, SEA plus JKO, or APAC minus China, he tells me. So that's 9 countries, Korea to New Zealand. He's also the author of the technical compendium on Streptomyces lydicus, WYEC 108, which just about sent him around the twist, apparently, getting all that information in the one place, but it needed to be done. That work pulls together 3 decades of published literature alongside a meta-analysis of, from what I can tell, 199 field trials spanning 73 different crop and pathogen combinations. So when Ross talks about what the evidence does and doesn't show, he's speaking from having gone through essentially all of it. and Ross is going to take us through the mode of action and the evidence base. We're also very fortunate to have Professor Dr. Vikineswary Sabaratnam with us, who most people refer to as Professor Vicky. Vicky is Honorary Professor at the Institute of Biological Sciences, Faculty of Science at the University of Malaya. She spent more than 3 decades working on Actinomyces and biocontrols with over 260 peer-reviewed publications to her name. She's the founding president of the Mushroom Research Association of Malaysia and a fellow of the Academy of Sciences Malaysia. Vicky also brings an independent academic perspective to biology, and we're grateful that she's joined us this afternoon, and hopefully she might contribute later on as well. So just a reminder, questions into the chat as they occur to you. I'll interrupt Ross. And Ross, if you don't mind, would you be able to take over and share your screen?
Thanks for the great introduction, Steve. I'd also like to acknowledge my co-presenter today, Professor Vicky. She and I have been working with Actinovate for the past 5 years on a particular disease of oil palm basal stem rot caused by Ganoderma. I'll make a few references to some of that work in the course of this particular talk. The slide deck, so while the video recording will be available, I'm also very happy to share the slide deck as a PDF file. Almost everything in here is in the public domain if you know where to look for it. So very happy to share the information, get people talking about it, try and encourage people to think about what it means in the cropping programs that you're executing. So our topic today, it covers a range of areas, including the mode of action and what evidence we have for the performance of Actinovate, certain use practices for how we utilise Actinovate in cropping systems, and just setting expectations around how this product can be used successfully and avoid disappointments that might have happened in the past. So Actinovate is a Streptomyces, Streptomyces lydicus. The strain is WYC108, so it's a gram-positive microbe. It was identified and characterized by Professor Crawford at the University of Idaho. He went on sabbatical to England, made a large collection of Actinomyces strains, took them back to the US and screened them, and then had a whole array of students working on characterizing WYC108. So it's a saprophyte. It lives off decaying material. And a particular feature is it's got the capacity to colonize roots, and the root colonization ability will be certainly a feature of this talk today. So this next slide is summarizing that history I was referring to at the University of Idaho. So Crawford went around fields in England. He dug up soil, sometimes just bulk soil, sometimes where crops were growing, and he isolated Actinomyces from either the bulk soil or from the rhizosphere of plants. And he went through a series of selection steps that led to the selection of WYC108. And one of the features of those selection steps is he specifically selected a strain for its antagonism towards Pythium. So it was coming with known antagonism to an oomycete. And in addition, he screened against 4 wood rot fungi, so basidiomycetes. And then he also selected for its plant growth promoting abilities. So all of these attributes, so antagonism towards oomycetes, fungi, as well as plant growth promoting abilities, were a feature in the selection of this particular strain. And there was really very detailed characterization undertaken by a whole range of students to establish that. Actinovate was first registered in the US in 2004 by a company called Natural Industries. They took a license from University of Idaho. So Natural Industries was the commercializing business, and Novozymes, a predecessor business to Novonesis, purchased Natural Industries business in 2011. And range of registrations are EU, Australia, New Zealand, Canada. Now, where I'm going to move to next is I'm calling this section biology refreshers, because I'd like people to be thinking in terms of the biology of the organism and the system in which it functions. I think this is something distinctive about biocontrol products in the way that we need to think about them and the way that they work. The modus operandi is not let's spray and kill the target. It's fundamentally about the biology of the system and how, through leveraging the attributes of the plant, the environment, the biocontrol agent, interventions we can make, how we can actually bring these things to bear to suppress plant diseases. So this particular slide is talking about the life cycle of a particular pathogen. So I grabbed our life cycle of Ganoderma bonanense. It starts with spore germination. It goes, because it's a basidiomycete, there's monokaryons produced. Those monokaryons have to mate before they're pathogenic. The mycelium will grow and produce a basidiocarp, and then a fresh range of spores. So throughout that cycle, there are many different points of intervention where potentially biocontrol agents can be effective. You can suppress spore germination. We've hypothesized that you can interrupt fungal mating. You can certainly disrupt mycelium development, and you can also disrupt production of the fruiting body. So there's not one point of intervention that can be achieved by biocontrol products. There's multiple potential points of intervention, and of course, this is one of the things that makes them very powerful as agents for controlling plant pathogens. So something else to consider is the specific nature of the pathogens that you're targeting. And here I'm talking about the particular cell wall characteristics. The cell wall characteristics of fungi are quite distinctive, and they're different from the cell wall characteristics of bacteria and oomycetes. So when we think about how a biocontrol agent can act as an intervention against pathogens that are either fungi, bacteria, or oomycete, you have to think about what the biology is and what the modes of action of the biocontrol product are to actually set you up for successfully controlling a particular disease. It's about the interaction between the biocontrol agent and the pathogen that you're targeting. And it's not one size fits all. There's a whole lot of nuances connected with this. So something else to consider is the response of the plant. And so the plant has an immune system, and ordinarily that immune system will be triggered to provide a level of resistance to the pathogen that may be present and otherwise attack the target plant. But what of course we know very well is that these pathogens have evolved quite remarkable ways of either suppressing the immune system of the plant or avoiding the immune system of the plant, such that that immunity is actually not effective against some of these pathogens. But what that means as well is that wherever we can actually reactivate that immune system, and so here I'm talking about products that induce immunity, and here I wrote it as Actinovate, which has a well-known mechanism of triggering salicylic acid production. In this particular case, we can reactivate the immune system of the plant to enhance its resistance and actually avoid the pathogenicity that would otherwise occur. That leads me also to say is that many biocontrol products have this capacity. A feature of them is that they will activate the immune system of the plant when otherwise it might have been suppressed. And then on this slide, again, in the context of biology and how we think about the interaction of a biocontrol agent, the pathogen, the environment, and where these organisms are operating, it's not just about the pathogen alone or the biocontrol product alone. There's many things interacting with one another at the same time. And so one of the things that we can be thinking about is, how do we promote the activity of the biocontrol product? So here on this slide, I'm calling it priming or waking up the actinovate to stimulate metabolite production. You don't just put it out in the environment and just say, good luck, hope everything works out okay. You need to set it up so that the actinovate is in a situation where it can perform to its maximum potential. That includes concepts like feeding the actinovate. It has an obligate requirement for carbon and nitrogen. Where's it going to get the carbon and nitrogen from? Again, don't just put it out there, hope for the best, and think that everything's going to be okay. Do things like stimulate the health of the plant, stimulate the immune response, as I was just referring to, use other biological products that are known to be beneficial and also can be supplying carbon and nitrogen. So there we might be thinking about seaweed or humic and fulvic acid, which are becoming more and more prominent in production systems today. In terms of best practices, always think preventative. While the products can work curatively, the timing, there is a vital aspect of introducing the products early in advance of disease development. Let them start producing their metabolites that have the properties that suppress the development of disease. Get them established, get them colonizing plant roots, get them living on the plant, get them producing the metabolites. So timing's critical. You also need to think in terms of the use rates. And I'd encourage people, rather than thinking about grams per hectare and grams per litre, start thinking in terms of CFU numbers per metre squared. So a common maximum rate you'll see for Actinovate on the label is 840 grams per hectare. That equates to 840,000 CFU per metre squared. So if you can think in these terms, what is the level of activity I need to have in the field to be able to accomplish a substantial level of suppression of disease organisms that may be present? So that 840 grams per hectare is 840,000 CFU per square metre. You can run it at higher rates, lower rates. You can use dilutions where you're fertigating and drenching, trickle irrigating, and so on. But you need to think carefully about what dosages you need to be applying to ensure there's sufficient inoculum there to be producing metabolites that suppress disease. Also thinking about adjuvants, the Agreva team can help you with adjuvant recommendations. just looking at that graphic on the left-hand side, what that's actually about, I'll touch on a bit later in the presentation. But this particular slide is about what happens when you add chitin flakes into the furrow with the Actinovate, looking at suppression of nematodes. And so in this particular study published in 2011, Berlin's paper, absolutely fantastic increase in the level of suppression of root knot nematodes in tomatoes when the chitin was added with the Actinovate. So my point is, it's not just about put the Actinovate out, hope for the best. You have to prime it. You have to feed it. You have to use the right rate. You have to think carefully about timing. It's not just throw and hope for the best. You have to think carefully about all these different dimensions, and all of it's related to biology. Just have to understand the biology of the system and leverage the biology of the system to your advantage.
Excuse me, Ross.
Yeah.
I've just got a question from a participant wanting clarification on one of the previous slides. He wanted to clarify whether it was saying that Actinovate does fungus but not Oomycetes.
Sure. Well, we're going to get to that in a second. There's good evidence for control of Pythium, Phytophthora, good evidence for control of Oomycetes, and we'll get to deal with that shortly.
Thank you.
Okay, so modes of action, and what I'm setting out here is fundamentally there are 4 modes of action. One of the best characterized modes of action is that Actinovate produces enzymes, particularly chitinase and beta-glucanase. The effect of those enzymes is to break down fungal cell walls. So earlier I was talking about the biology of the pathogen. So we know that fungal cell walls are made of chitin and beta-glucan. So the production of chitinolytic and beta-glucanase enzymes by Actinovate has the effect of parasitizing the fungal cell walls, leading to degradation of the fungus. And again, I'll actually show some pictures of how that happens shortly. The second mode of action is there are some really distinctive metabolites produced by Actinovate, and those metabolites have various effects on fungi, bacteria, and oomycetes. The metabolites are different. The metabolites that act on fungi are different to the metabolites that act on bacteria and oomycetes. So there's a range of metabolites, and those metabolites act on fungi, bacteria, and oomycetes. Another key feature, I spoke briefly before, Streptomyces have the ability to colonize plant roots. It is a strain-specific characteristic, and it differs according to the plant species. So depending on the strain, depending on the plant species, you'll achieve certain levels of colonization of plant roots. That has the effect of competing for pathogens that are in the soil. It deprives them of nutrients. There's less space available for pathogens to invade plant root systems. So that ability to colonize and basically keep out, exclude pathogens is a very important attribute. But as I say, it's a crop-specific attribute, and is actually a strain-specific attribute of Streptomyces. And then the last characteristic I was going to talk about is the ability to enhance plant growth. So when Crawford selected WYC108, he identified in growing lettuce that he could enhance the growth of lettuce by inoculating the plant roots with this strain. And later this was characterized, connected with siderophores, Again, I'll have a look at this in a bit more detail in a few minutes' time. So multiple modes of action, doing different things, all of it to enhance the health of the plant, exclude pathogens, degrade pathogens, so to sustain the health of the crop.
Ross, just going back to the nematode
Yep.
one,
Yep.
we've got a participant was asking whether it was nemastatic or nemacidal.
I'm fairly sure the paper says nemacidal.
Okay.
So yeah, and I think I've got that on the latest slide as well, nemacidal. Mm-hmm.
So we can look up the we've actually got a copy of that paper somewhere, haven't we, that we can supply?
Yeah. But I think the answer, I'm quite confident the answer to the question is nemacidal. Mm-hmm.
Thank you.
Yeah. Okay, so this slide is just emphasizing these different metabolites produced by this Streptomyces strain. It's producing an antibiotic. It's producing multiple antibiotics, actually. In the case of fungi, we know that the natamycin binds with the ergosterol in the fungal cell wall, and by doing that, actually disrupts the membrane inside the fungal cell wall and leads to disruption of the growth of the fungal mycelium. For bacteria, there's a different antibiotic that's important. Streptomyces is important. In the case of oomycetes, not very well characterized. The PhD student that Crawford had working on how WYC controls oomycetes, the PhD student was inconclusive in his findings. He wasn't actually able to determine what was the cause, what was the molecule that was disrupting the pythium. So we're speculating here that it's this molecule that I put there disrupts biotin synthesis. But of these three, this one is the one that is most uncertain. But the point here is there's multiple modes of action that give this relatively broad range of activity across fungi, bacteria, and oomycetes, as well as nematodes. Okay, persistence in the rhizosphere. It's really a key question. What happens when you use this product? You inoculate a plant. What you want to achieve is the establishment of the biocontrol agent on the root system on the plant, such that it lives off the root exudates. It's benefiting from the carbon and nitrogen and sugars from the root exudates. And CFU counts have been done from several different studies for a period after the inoculation event. So Yuan's PhD thesis, he measured 10 to the 5 CFU per gram after 30 days. The Couples paper working on tomatoes, she measured more than 1,000 CFU per grams more than 2 weeks after inoculation. There's a really critical aspect. You want to get the Actinovate established on the plant, living off the plant roots, producing its metabolites. And so the point underneath that, in the non-rhizospheric soil, if you just put Actinovate in bulk soil without plants being present, it will persist for a maximum of 100 days, after which it basically degrades because it doesn't have an adequate, in most cases, not going to have an adequate supply of carbon and nitrogen to survive for a long period. So really critical topic here about its persistence in the rhizosphere. Again, plant growth promoting effects. So I was talking about siderophores, and I pulled the pictures out of Tocqueville's paper. So he worked on pea plants, and so you can see it here in the picture in the middle panel. Pea plants at 30 days, much enhanced growth of pea plants at 30 days. The one on the left, labeled A, inoculated with Actinovate. The one on the right is the control. So a really distinctive plant growth promoting effect. He also was looking at the bacterioid activity in the root nodules. And so the picture over here on the right, he was actually demonstrating much enhanced bacterioid activity in nodules of plants inoculated with Streptomyces lydicus. So really interesting area, not well developed, I would say, in the science sphere. What is actually going on in these systems and what role does Streptomyces have in plant growth promoting? It's not just about the protection of plants, it's also about plant growth enhancement. But there's certainly opportunities to do a lot more good work in these types of areas. So getting to what's already been tested. So over the past 30 years, and since Crawford's work started, as I say, he went on a sabbatical. He published a paper in 1993 that sort of introduced WYC 108 to the world. So over these past 30 or so years, there are many scientific publications, more than 30. There are, by my count, 16 PhD and master's theses, 7 of which come from Crawford's group, University of Idaho. So a lot of published evidence, and then the Novanesis database. There are about 400 trials, more than 400 trials. I was quite selective pulling data out. I wanted to be sure that what I was retrieving out of different reports, that I was able to report the data accurately. It included where Actinovate did not go very well as where it did go well. I put in everything where I could be confident about the data. So this became the background for the data set that was compiled, and which we'll go on and have a look at. This is just talking about the post-commercialization graduate research. So there have been at least 9 theses by my count. It's been quite a fertile area for student activity, where people have been interested in biocontrol products. Studies on all those different crops that I'm mentioning there: tomatoes, grapes, some really, really good work. 2 excellent theses in the US on grapes, among others, strawberries and watermelons and tomatoes and so on. So now, how do we look at efficacy of a product like this? And so there's really 3 areas we can be thinking about. So first of all is the antagonism studies that we can just do on a benchtop in a lab, and we can learn a lot by that. But it doesn't tell me will my Actinovate colonize the roots of my crop that I want to protect from disease. And it doesn't tell me what actually happens under real disease pressure. I just learn about the interaction between the pathogen and the Actinovate, and I can particularly learn about useful use rates for the Actinovate. Of course, pot and greenhouse studies. So where Vicky and I have been working the last 4 years or so, everything in the lab, everything in the greenhouse, trying to learn as much as we can, controlled environments, understanding the modes of action, what you need to do to get to maximize what the level of disease control that Actinovate can achieve. And then finally, field trials. As I say, there's been we have a database with more than 400 trials reported. We've been through those data, cleaned it up a bit, and so today we're working with this data set. It's about 199 trials, and there's a lot more information to extract out of it yet. These are somewhat early days, analysing the data and reporting it out like we're doing today. So I'm going to go to the antagonism studies that we've been doing, and Vicky might chime in here and help me. But with these antagonism studies, we'd like to use the next 3 slides just to illustrate some really important points for how to conduct these studies. So in this particular case, we were looking at Actinovate, controlling Fusarium wilt of banana. And I'm sure many of you are aware Fusarium wilt is a global pandemic. It's in Australia, in Northern Australia. It's pretty much global now. It's a major threat to Cavendish bananas. So we looked at what's the level of activity for Actinovate against both race 1 and the tropical race 4. Tropical race 4 is the big threat at the moment. So what we do here in studies like this, the panel on the right-hand side, on the left-hand side, this is against tropical race 4. And the bottom row is where is the control. There's no Actinovate here. And we're looking at these plates 9 days after establishing these studies. So what we do is we have a control plate and a test plate, and we do cross plugging. So we grow the Actinovate, we grow the pathogen, and then we transfer the pathogen to a new. We transfer the Actinovate to a plate. We transfer the pathogen to the plate with different numbers of days between introducing the pathogen following the biocontrol agent. So if there's only a 3-day gap between introducing the pathogen after we've introduced the control agent, what we see here is that the pathogen completely overruns the plug of Actinovate. The Actinovate is not able to control the pathogen, and the pathogen here in the middle completely overruns the Actinovate, which is lawned on the bottom side of the petri dish. If we go to 5 days, the Actinovates had more chance to produce metabolites, the antifungal metabolites that suppress the disease. So instead of seeing the pathogen overtaking the Actinovate, now we see a nice halo where the pathogen is constrained within this small region. The Actinovate is producing its metabolites, it's suppressing the development of the disease. And if I give the Actinovate a 7-day head start, so it's been sitting here, it's lawned, it's actually producing the metabolites, and we can see very substantial suppression of the plug of Fusarium in the middle of this petri dish. So here we establish a very important point, something I was introducing earlier, that it's very important to get the Actinovate established early. Let it produce its metabolites, let those metabolites suppress the development of the disease as that disease is initiated and tries to get established. Over here at 3 days, if you co-cultivate or you don't give enough lead time, you'll get completely overrun. Whereas if you give 5 days or 7 days, you can see reasonable levels of control sufficient to suppress the development of the disease. So we had the same pattern in with the race 1, and so it's these types of studies that let us understand something about the dynamics of the disease, the way the pathogen and the biocontrol agent interact with one another. It does confirm if you don't if you don't give the Actinovate a head start in aggressive diseases like Fusarium oxysporum, then it can well struggle.
So, Ross, would that be the same for foliar diseases, for instance, in countries where they can use it on grapevines? Would they be well advised to go earlier prior to?
Definitely go earlier. And there's a master's thesis from California, and the student's name starts with M, but I'm probably not going to do any better than that. He shows this very clearly, very clearly. Put it on early, don't wait.
Yep. Thank you.
There's actually lots of evidence in the literature showing that you should apply early before the disease significantly develops. It's just that it's scattered, it's buried. And of course, the opposite also exists. We're looking at one particular crop recently, went to find the published data. Is there published data for Actinovate controlling that disease in that crop? Yes, there is. But in the study, they inoculated the plants, let the inoculated plants go for 7 days in the glasshouse before they treated with Actinovate. And of course, what happened was all the plants got clobbered. Basically, everything got 100% disease. So that's the opposite, right?
Yeah.
You have to get it established early. You have to support it. You have to prime it. You have to get it going. You have to get it producing the metabolites. And I think this is a universal thing with biocontrol products. This isn't really unique to Actinovate.
Thank you.
So just another example what we use these antagonism studies for. So in this particular case, I'm returned to our basal stem rot work with Ganoderma bonanense. And so over here, I have a plug of the Ganoderma in the middle of the plate. I have a well of Actinovate, which was added at 0.1 gram per litre. This well here was 1 gram per litre. And this well up here, the third well, 10 grams per litre. And so you can see over here that this lower dose rate at 0.1 gram per litre, it's actually insufficient. There's not enough production of metabolites to suppress the development of the pathogen, which is growing out from the middle of the dish. On the other hand, when we get up to the 1 gram per litre or the 10 gram per litre, they're producing metabolites that lead to the suppression of the disease. And then something else that we can do, we'll grow the pathogen, cross-plug it over here. And then here we've got the Actinovate lawned on the bottom side of the plate. And then we're looking in this zone here where the red arrow is. And then particularly in the box here, we can actually take the agar and then do scanning electron microscopy on the mycelium that we find in this zone between the Actinovate and the pathogen. And so when we do that, we'll actually find deformed hyphae, fragmented hyphae that's lysed because of we presume through the action of those chitinolytic and beta-glucanase enzymes. So these sort of studies again can really help us establish what the Actinovate is actually doing in the way that it interacts with the pathogen. Those modes of action I was talking about before, we can actually visualize them in practice through just some benchtop studies like this, growing some pathogen and the Actinovate, and then harvesting at this frontier. We can actually learn a lot.
So, Ross, if we prime the Actinovate with chitin, does that improve the inhibition side of it? And is there any mutational studies to identify the exact molecules involved?
Yeah, so Vicky and I have done a lot of work on priming, and the Bel Air study was really distinctive in the way that there was an advantage when you added chitin flakes with the intent of supplying carbon and nitrogen to the actinovate. When we've tried to reproduce this, hopefully Vicky might make a comment, we haven't been so successful, and we've been looking for different ways of priming. It's not just a simple matter of putting chitin flakes in. We tried that and we didn't really see a distinctive advantage in our oil palm studies. So yeah, I think it's a really interesting area. I think it's a really important area. It deserves much more attention than has been given to it today. Do you have any comments on that, Vicky?
She's still here. No, she's still here. She's on mute.
Okay. Let's keep going. I think the points the key points are well made, and Vicky can join us for some from Q&A later on. So I was talking about we're thinking about how to characterize the performance of a product like Actinovate, biocontrol products in general. So the antagonism studies are important, but now we get to the field studies. Someone's already asked me about field studies. So one of the things we did was we were trying to categorize the responses in different pathogen by crop combinations. So remember, I had about 199 trials. I can't remember, it's like 70 or 80 different crop by pathogen combinations that were studied. And so we needed some sort of language to be able to define what sort of responses we were getting. Trivial response when I don't reduce the disease by more than 10%. A modest response, 10 to 40% level of reduction in disease incidence or disease severity. Substantial level of response, 40 to 70, and very strong response, 70% in disease reduction. But the other thing that we were interested to think about is one reference point is obviously the negative control. It's something that hasn't been where disease control has not been implemented. So you can compare it to the relative control. But the other important question is, how does it do relative to the best available chemistry that was included in the trial? So here we had these criteria. Actinovate significantly better, so it actually performed better than the synthetic chemistry in the trial. It was equivalent, or we can't distinguish between synthetic chemistry and the biocontrol. Inconclusive, so it's starting to get, there's a gap where we might be saying, well, it's not conclusive. And then finally, the standard where we're using synthetic chemistry is better. So we're thinking about all these different things and trying to get a consistent language about how to report these results. And what we're going to look at is forest plots. Strictly speaking, I avoid calling this a meta-analysis because strictly a meta-analysis has a few requirements that can't be met with these data. So I don't call it a meta-analysis. It's just a aggregation of the results using statistically valid method to visually summarize particular crop by disease combination results. So we'll have a look at each one of these individually, but what you're going to see is we've got a set of results for crop by pathogen for fungi, crop by pathogen for oomycetes, and crop by pathogen for bacteria. And so there's in our data set, there's 49 combinations from 109 trials of fungi. For oomycetes, there's 13 combinations, and for bacteria, there's 10 combinations. I'm not showing all the fungi combinations. I'm just going to show you one graph for illustration. But if anyone's interested to see what else is there, I'm happy to send the graph. So this is what we've got for fungi. And in this particular case, I filtered so that we have at least 2 trial results for a particular crop by disease combination. So cucumber, mildew, at least 2 trials. This dark colored dot is actually the synthetic chemistry performance. So we got a level of disease reduction versus the untreated control, round about the numbers 65%, something like that. In these 2 trials, actually the Actinovate does better. So in cucumber with this mildew control, the Actinovates are doing better than the synthetic chemistry. And then when we come to the next one, strawberry, the synthetic chemistry achieved a high level of disease control, and the Actinovate did really well too. It's not really very far behind. For botrytis with grapes, the Actinovate did better than the synthetic chemistry did. And then you can see this sequence where the level of disease control achieved by the Actinovate, it diminishes as we move down through this particular crop by disease combinations. But nevertheless, you see results like this on potato with blight, apples. The synthetic chemistry didn't do very well, and the Actinovate did almost as well as its synthetic. So we're able to sort of categorize these results according to the relative performance of the Actinovate, the performance of the Actinovate relative to the best available chemistry, and reach some conclusions about what is the general pattern, how well Actinovate's doing for controlling fungal disease. I wanted to point out this one for the potato, again, where the Actinovate does better. In fact, those trials, those 7 trials, all of them had quadris in it. So in all 7, on average, across those 7 trials, we're beating the Strobel and synthetic chemistry product. So that's pretty impressive. 7 trials and we're beating the synthetic chemistry. So, you know, this is starting to illustrate what the range of capabilities of this particular product is. And then we can go to bacteria, Xanthomonas, which you'll get on range of crops from walnut to tomato, and everything you'll see is down here, tomato with Xanthomonas. So we can get reasonable levels of control, and actually something reasonably equivalent to best available chemistry. One of the things to remember with bacterial diseases, though, is people were often using copper. So most of the time, the synthetic product here is copper. And so any of these situations where the biocontrol product is, I'm not saying it's the same as the synthetic product, but doing pretty well. Again, 9 apple trials, fire blight. It's not too shabby. The level of control, 75% disease reduction in apple, and the Actinovate achieved 50% averaged over 9 trials. So it's not too shabby. It can has an effect on these diseases, and we can achieve commercially important levels of disease control. Then moving to the next one, the oomycetes. Remember that Actinovate was selected for its antagonism towards Pythium. So some of these ones up here at the top, unfortunately, it's only just 1 trial on carrot, but it did better than the synthetic chemistry. Again, Pythium on geranium, it's equivalent to the synthetic chemistry. Then we get into Phytophthora diseases, doing really quite well here. We've got bits of gaps here. Lettuce, cucumbers, it's doing as well as the synthetic chemistry. So again, what we saw, reflecting on the sequence, fungi, useful control across a range of crops and diseases. Bacteria, useful control against a range of crops and diseases. Oomycetes, control against a range of crops and diseases.
Hey,
And
Ross,
yep.
No, you're right.
Yep. Go ahead.
When you finish that one, we've just got a few questions to get through before you move on.
No worries. So just summarizing those results, I had the categorization at the beginning, and basically I used it to give a pass or fail mark, pass or fail. So in the bacteria combinations, fungi, oomycete combinations, how many crop by disease combinations where the pathogen is a bacteria does it fail versus how many does it pass? Fungi, crop by disease combinations, pass, fail. So I gave it these marks. Bacteria, okay, half the time it's achieving a useful level of disease control. Fungi 40% of the time, oomycetes 40% of the time. It does tell you that you need to be thinking carefully about how to use the product, which crop by disease combinations are actually target. But this is actually a product that does have quite broad efficacy, established on the back of that range of modes of action I was talking about earlier. It's not a universal product. It's not paraquat killing weeds. It has the ability to colonize hosts. It has the ability to produce metabolites. It has the ability to suppress disease. But it's not universal, but you can set it up to get really important results across quite a wide range of situations. I think now we're good for a break, unless I just do the nematode one. Nematode one's an orphan slide. This was just saying, let me finish this one. This is just saying two important studies, Belair, Dillon, that really highlights the chitinase production suppresses the production of juveniles from nematode eggs. The nematode eggs are made of chitin. The chitinase degrades the wall of the nematode egg. You get fewer eggs, you get fewer juveniles, and in addition to that, someone was asking, nematicide, is it nematicidal or nematostatic? It's producing, the evidence is it produces a range of metabolites that are nematicidal, causing toxicity and mortality. So this is a place to stop, Steve. So I did fungi, bacteria, oomycetes, and nematodes.
Okay, no worries. So we had a question overhanging with regards to the plating that you were doing. And one of the participants wanted to know if there was any mutational studies done to identify the exact molecules involved. And that's
okay. Yep. The origin of this work is Crawford's lab. Crawford retired. His last thesis student was 2006. So we've now got a 20-year gap between anything being done in this area. So unfortunately, whatever exists is published in the papers that came out of the labs, those theses from Crawford students, and essentially nothing since.
Sound like a good job for a few more students. So next one is from one of our potato colleagues. She'd like to know whether the visual disease, whether the data from the disease suppression was done by visual inspection or observation or DNA levels. I gather that the agent, most of the research would be visual, wouldn't it?
Yeah, hi Audrey. All of this visual, all of it visual.
Yeah. And then one of our CSIRO friends is asking whether each crop pathogen trial, were they conducted at the same or different field sites? I reckon we have to do it.
Yeah, these are trials from many different countries and places at different points of time.
Yeah, I think that might be one of the one. I think Louise might be one of the people we could forward our that work he did on that compendium. We might be able to forward that to Louise.
The big file, the 110 megabyte file. All right, no problem.
And then there was a question about how many applications we used on the potato crop, but as you say, it was probably multiple trials, but I gather most of the potato stuff would have been done in furrow from what I can tell.
Yeah, look, I think for specific questions like that, the better thing is just dig out the data and the papers, because there's going to be a range of things, and I don't want to guess in the big body of literature. So just on potatoes, there was a really nice thesis from Michigan on verticillium, published 2024. It's got some super interesting results in it. So yeah, there's ongoing work. Um, yeah.
All right, you can send that to me and I'll forward it on.
Yep.
This one here, water pH affect the activity of Actinovate?
I'm sure with so Actinovate can be, it dissolves in water. It's used as a drench in fertigation, sprayed, uh, and it's essentially insensitive to pH between. I'm fairly sure our data is 3 to 10, something like that.
Yep.
Can I just jump in? Can I just jump in then? Just chlorine is an issue with Actinovate, though.
Yeah. So one thing to watch out for is there's a low tolerance on the presence of chlorine. Town water would generally be okay, but we do recommend not using chlorinated water. And we do have suggestions for how to dechlorinate water if you don't have a unchlorinated water source.
And what are the key environmental constraints that means the Streptomyces will work in one situation and not the other?
Yeah, the vital thing is to get it colonizing the plant roots. You have to get it established on the plant, living off the if we're talking on the root system, living off the exudates from the plant roots, using the carbon, using the nitrogen. Now, if you don't get it established well, and I mentioned before that the crop species is a factor, if you don't get established well on the plant, then it's going to struggle. And I think this is actually probably the biggest thing. You have to actually introduce it in a way that lets it get well established on the plant roots.
Food source, right. Okay, doke. That's got us cleaned up on the question front, I think, for now.
All right. Just a small addendum to that. One of the things that we've learned in our oil palm studies, so with Ganoderma, is that relative to other Streptomyces species that Prof. Vicky has worked with, WYC 108 is actually what we call a shy colonizer of oil palm roots. Other Streptomyces species much more readily inhabit, colonize oil palm plant roots than what Actinovate does. It's what the term I use is a shy colonizer. So don't just assume that you can put it there and it's going to colonize. It's definitely a key consideration. Am I going to be able to adequately get this established on the plant and get it to do what I need it to do? Don't just think that that's a given, because depending on the crop and, as the question referred to, maybe the environmental conditions that are prevailing, it may not necessarily do that. And we were looking at different ways to try and enhance that colonization. I would say without success. And we attributed it to the talking that goes on between the plant and the Streptomyces.
Mm-hmm. Okay, thank you. So we've got a participant here that believes they've got high chlorine in their soils and they're looking for a chlorine ion concentration to avoid.
Maybe we have to take that offline, because can't do it off the cuff.
No worries. And I don't know whether you're going to cover it in the rest of the thing, but someone's asked with regards to whether it plays nicely with other beneficial organisms such as trichoderma, and I guess the other one would be bacillus.
Yes, it does. They'll coexist really quite happily.
Then we've got a question here about whether it's been used as a seed treatment, which I think is correct. It has and does. And I think that
so you
question
can dress seed with it, certainly.
And I think that's from what I can tell, that's a pretty good way of using it because it establishes early. Yeah.
Yep.
It's actually on the label too.
Yep.
Yeah. That's how we use it in oil palm. We actually treat the seeds.
And I can answer that one. Okay. I'm ready to go. We're 57 minutes in.
Yep, and I think everyone, we're in the home straight now, so just bear with me for a bit more. Let me finish getting through this with some take homes. Use practices. So I've made all of these points before, bringing them back again. Always favour early inoculation. It needs time to colonise, start producing metabolites. And this is actually known in the literature, but you have to know where to look in the literature and to be able to perhaps properly appreciate just how important this is. It probably performs better under moderate disease pressure rather than high disease pressure. And again, that's just connected with it does have a finite capacity to suppress developing disease, let alone disease that may already be present. So this is talking about matching the application method to how you want to protect the crop. The colonisation quality, very important determinant of the persistence. I've been making that point numerous times. Works best on certain hosts, pea, soybean, tomato, cotton, linseed. I would put our oil palm in a shy colonizing group of plants. Preemptive timing, avoid poor colonized, so actually lettuce. It's actually used on lettuce, but I wouldn't characterize its colonization of lettuce as being something that's very promising. It only colonizes lettuce reasonably well. Foliar application, that sort of colonization is irrelevant, but it still needs to get established on the plant. It still needs to get carbon and nitrogen from somewhere. It does mean that you're looking at sort of weekly application intervals, and doing that, it's really quite well established that you can achieve significant levels of disease control for diseases like botrytis and powdery mildew in grapes. This last one is the master's thesis from California. That particular one really establishes how you can use the Actinovate in concert with synthetic chemistry products, use it in rotation and achieve really good levels of disease control. So choosing the right target, those graphs I was showing before, there are certainly crop by disease combinations where we don't have strong supporting data for the efficacy of Actinovate. On the other hand, we do have relatively clear data, potato, rhizoctonia, pythium, and carrots, and so on, where Actinovate is a really good fit and can be an important part of production systems. So this is, again, just referring back to what I was showing before, the graphs, just transferring it into some words. This is where it works reasonably well. This is where it doesn't work so well. So this is preventative treatment. Make sure there's adequate level of inoculum, make sure the root colonisation and enhance the plant health. Those things I was talking about before, seaweed, humic, maybe use a immunity-inducing product. All these things can really help Actinovate's efficacy. By the way, in our data set, I mentioned all the trials, 400 trials. We do have data where ActiGuard was used with Actinovate across quite a large series of trials. A future version of this talk will actually summarize those data. I just haven't quite had the energy to put it all together just yet, but eyeballing it, Actinovate and ActiGuard is actually pretty impressive combination. So setting realistic expectations, I like to talk about Actinovate being a rotation partner. It's not intended to be a replacement for synthetic chemistry. If you really want to protect your crops from disease, you need to be thinking about integrating the biocontrol with the other practices you might already be using. Give some relief to make a contribution to the resistant management strategies. Good performance against a range of bacteria, quite good performance against a range of fungi. So you can find a fit for this product. I think I'm now really starting to get to repeat myself. Yeah, why rotation is important, resistance management, where the trade-off is favorable. Last slide, the mechanisms. There's a whole range of mechanisms. I talked about 4 mechanisms. Evidence, there's actually loads of data. There's no need to, in my view, repeat a lot of the things that I've already done. Just need to go and find it. Everything I've shared today, except for our Ganoderma work, is published. The measurement methods you need in Novanesis, we've really benefited so much in our work from working with Prof. Vicky and her team at the university. The mycology skills have really helped us solve key aspects of how we control a disease like Ganoderma boninense. Those mycology skills, working alongside agronomy and plant pathology, really, really vital. Use practices, what diseases you're targeting, and just set it up as a resistance management rotation partner is normally where I'm going to be recommending. It's not in that space exclusively, but it's where I like to advance positioning this product. That is the end. So we should have a few minutes left for some more questions.
Did you want to unshare your screen, Ross? I'd just like to thank you very much, Ross, and Vicky. I'm not quite sure what happened to Vicky. She was here and then she went, but that's okay. So I think you've covered it off quite well. There's probably a lot more questions that folks have got because of the format that we've set up. But as I said, we've recorded this and we'll share the link around so you can replay it backwards and forwards. But also, if there's any questions, any burning questions, you can ask them now, or you can email me and we'll connect you up with Ross, or we'll bounce it off Ross backwards and forwards. And I also think there's probably some people that have specific questions about specific trials, in which case we'll probably have to drill back into your trials database and seek those particular questions out. So that being said, thank you everybody for attending, and I hope you all got something out of it. And yeah, watch this space.
Thanks, Ross.
Okay.
Thanks, Ross.
Thanks, everyone. Bye.